Hello, dear children. A very good afternoon to all of you. So, how are you? I hope you all are absolutely fine. So, I welcome you to another class of 9th biology. And as you know that we have already started our journey of discussion with the cell organelles, right? So, just before moving on to next new further topics today, first thing, as I told you in earlier class also, the moment you join any live class, please mark your attendance by writing your 5-digit code, roll number, and your full name so that we can mark your attendance easily and clearly in our registers. Right, students? And children, yesterday itself, that is the portion of the chapters, various chapters in science which has been removed is already posted in a school group. I hope you have already made a note of that. अगर नहीं किया है तो उसको नोट करेंगे. अब इससे पहले जो जितना पढ़ लिया that's no problem. But now in further chapters we would be dealing only with those topics which are prescribed by the CPSC for this present session, that is 2020-21, right? So students, in last class regarding cell organelles, I discussed with you first that what are cell organelles. And there we learned that cell organelles are the double membrane bound structures which are an important component of an eukaryotic cell, right? And then we started about the discussion of various cell organelles. And first I discussed with you about endoplasmic reticulum. And there we also learned about two types of endoplasmic reticulum, that is smooth endoplasmic reticulum SPR and rough endoplasmic reticulum, that is RER. And we discussed the differences between those two, right? And major difference was that in appearance and function, that SCR it appears smooth under a microscope and RER it appears rough under a microscope, because on the surface of rough endoplasmic reticulum attached ribosomes are present, right? And main function we say that a smooth endoplasmic reticulum it synthesizes lipids, whereas rough endoplasmic reticulum it basically synthesizes proteins. That's quite obvious because on its surface what are present? Ribosomes, and ribosomes are nothing but the protein making factories, right? Then we discussed about Golgi apparatus. There we came to know that Golgi apparatus is the packaging house of the cell. And then in the end of the lecture I discussed with you about lysosomes. Yes, the suicidal bags of a cell, right students? So in today's lecture we will discuss a bit more about some more important cell organelles, and in that sequence the first which I am going to take today is that is mitochondria. That is mitochondria, right? So student, mitochondria first, that is it was discovered by Kolliker. The scientist name was Kolliker. It was discovered by Kolliker, and the biologist name that is C. Benda. Yes, C. Benda. He coined the term mitochondria for this cell organelle. So sometimes what happens that children they get confused. So there is a clear clarification that discovery of mitochondria was made by Kolliker, but the term mitochondria it was proposed by the scientist C. Bender. So don't get confused, right? So now mitochondria as per structure wise, we say that it is a double membrane bound structure. It's a double membrane bound cell organelle. Like nucleus. Nucleus also has got two membranes, right? In the similar manner, mitochondria also has got two membranes. Another thing that in appearance under a microscope, it looks like that is sausage shaped, or sometimes that is biologists also describe it as rod shaped structure. Rod shaped cell organelle, right? Now regarding structure, it has got two layers. Now outer layer, the outer membrane of mitochondria is smooth, but it is highly porous. Means जो mitochondria की जो outer membrane है that is smooth और उसके सरफेस पर बहुत सारे छोटे छोटे पोर्स होते हैं. So we say that the outer membrane of mitochondria is smooth and highly porous. Means it allow different kind of materials to pass it through it down, right? So like that over the surface, tiny pores are present, right? So outer membrane is porous and smooth. Whereas the inner membrane of mitochondria, it is not smooth. It is not smooth because it is deeply folded inside the cell organelle in the form of finger-like projections, and these projections are known as cristae. These projections are known as cristae. So singular is crista and the plural goes cristae. Cristae. It is what? Cristae. Plural is cristae, singular is crista. So, inner membrane is deeply folded into finger-like projections, which is known as a cristae. Right? Now, the ground material of mitochondria, it is made up of a dense jelly-like material, and that material is known as the matrix. So we say that the floor of the cell organelle mitochondria, it is made up of a dense jelly-like substance which is known as the matrix. It is known as a matrix, right? And these finger-like projections which we are referring as cristae, these are basically embedded in the matrix of the mitochondria. And do remember, the inner membrane is not porous like that of the outer membrane. Students, in examination, sometimes we get a question, and twice or thrice we have already got in previous examinations also, that differentiate between the outer and inner membrane of mitochondria. So outer membrane is smooth, means it is not having any kind of folding into structure like that of cristae. It is totally smooth and it is porous. Whereas the inner membrane, it is smooth. It is smooth. It is not smooth, but and it is not porous, right? So that is a difference. Okay. Now when we see these cristae under microscope, they look as studded with dots. जब हम क्रिस्टे को माइक्रोस्कोप के अंदर ऑब्जर्व करते हैं तो उनके ऊपर हमें डॉट डॉट जैसे स्ट्रक्चर्स मींस इट appears as if they are granular by nature. So what happens over these particular structure finger like projections called crista, what happens are present small bodies which are known as oxysomes. They contain the particles called oxysomes, which are also known as F0, F1 particles. These are also known as F0, F1 particles. Another name for oxysomes. F0, F1 particles. So here this F, it stands for the name of the person, that is Fernandez Morales, who discovered these particles for the first time, F0, F1 particle, or simply oxysomes, right? Now these oxysomes are very important. These oxysomes, they play a very vital role. If you see in the structure of oxysome, that is, so this is what we say head, this is called stalk, and this part is known as the base piece. Yes. You see the structure of one single oxysome. It is made up of three parts: head, stalk, and base piece. Right? Now the head of this oxysomes, it is quite important because it contains ATP generating enzyme, and that enzyme is ATP, ATP synthetase. That one is ATP synthetase. It is the name of the enzyme, and that enzyme is present in the head of the head of the oxysomes. Where are oxysomes present? Over the cristae surface, right? Now this ATP synthetase, it helps in the generation of the particles called ATP. Now the term ATP stands for adenosine triphosphate. Adenosine triphosphate. You should remember this particular term very clearly, because asked in examination that what do you mean by ATP. So student, that ATP, it is referred as the energy currency of cell. It is the energy currency of cell. Means whatever energy is used or produced inside a cell, that is measured in terms of the molecules of ATPs, right? So it is known as adenosine triphosphate, and these ATPs are produced by the enzyme called ATP synthetase, which is present inside the mitochondria. It means the main function of mitochondria is to release energy. Yes. So that is the main function of mitochondria, to release energy. And children, in 7th standard, you have learned about respiration, types of respiration, aerobic and anaerobic, right? So do remember that aerobic respiration inside a cell always takes place inside mitochondria. Yes, the site for aerobic respiration inside a cell is mitochondria, and because of that, energy is released, right? And apart from releasing energy for the cell, we say that the main function of mitochondria is to release energy. Apart from that, mitochondria, it also help in the synthesis of amino acids. Yes, to some extent, mitochondria also help in synthesis of amino acids for the cell, and they also help in regulating the calcium ion balance inside the cell. Yes, सेल के अंदर कैल्शियम आयंस के बैलेंस को मेंटेन करने में माइटोकॉन्ड्रिया बहुत हेल्प करता है, राइट? But reference to your NCERT, the main function of mitochondria which is given is energy generation. That whatever food we eat, that food is digested and finally converted into glucose, right? After that, this glucose combines with oxygen to release energy, the same process which we call respiration, and that respiration takes place inside the mitochondria. And because of that, energy is released. Right? Okay. Now another one is another thing, that inside mitochondria, as in the diagram you can see that I have shown some ribosomes also with that blue chalk. So in the matrix of the mitochondria, you will always observe some ribosomes and a fragment of DNA. Now means mitochondria is having some ribosomes. It means it can synthesize its own proteins, right? It can synthesize its own proteins and DNA, right? As you might have learned in your previous lectures regarding the role of nucleus, that DNA is referred as the hereditary material of the cell, right? So because of that, this particular DNA and presence of ribosomes, that is mitochondria, they are capable of making their own copies. To some extent, they can replicate and make their own copies. It means some of their proteins they can synthesize even without the help of even without the help of the nucleus of the cell, right? And because of this reason only, mitochondria is known as semi-autonomous cell organelle. Semi-autonomous cell organelle means which can perform some more important activities of its own body itself, means without the guidance or without the help of the nucleus. Not all activities, but few activities. Like some of proteins it can synthesize, it can increase its population by multiplication to some extent. That's why it is referred as semi-autonomous cell organelle. So that comes one of the important term for examination, especially for a competitive part. Question is asked, why mitochondria is referred as semi-autonomous cell organelle? Because it has got ribosomes and it has got DNA fragments. And because of these two extraordinary things in its matrix, it can synthesize its own proteins and can replicate its number of copies to some extent. So we call it as a semi-autonomous cell organelle, right? So that is about mitochondria. Now mitochondria, since their main function is to release energy, right? So they are referred as powerhouses of cell, or these are also referred as storage batteries also. Yes. Now since mitochondria they release energy in the form of ATPs and this energy is used by the cell for various activities. So we say that mitochondria are the powerhouses of a cell, or these are also known as storage batteries of the cell, or these are sometimes also referred as ATP mills. Yes, इनको हम ATP mills भी कहते हैं. That is for mitochondria only. Okay, that is okay. So here from Parth, one doubt is there, madam. What is perimitochondrial space? Okay. So I told you there are two membranes of the mitochondria, outer membrane and the inner membrane, right? So the space which is present between these two membranes, that space is known as perimitochondrial, that is space, right? Okay? तो जो आपकी क्रिस्टे है, तो जो आउटर मेम्ब्रेन है, इसके बीच की जो स्पेस है उसको हम कहते हैं पेरीमाइटोकॉन्ड्रियल स्पेस, राइट? सो दैट इज अबाउट माइटोकॉन्ड्रिया. तो इसके जो कुछ कॉमन टर्म्स है याद रखेंगे, सेमी ऑटोनोमस सेल ऑर्गेनल एंड पावर हाउसेस ऑफ अ सेल. थर्ड वन इज एटीपी, फुल फॉर्म ऑफ एटीपी. Why it is called energy currency of the cell? Because in mitochondria, by the process of respiration, energy is released and it is stored in the form of ATP molecules, right? तो जब भी सेल को एनर्जी की जरूरत पड़ेगी, वो ATP molecules को ब्रेकअप करती है और सेल्स को यूज करती है, एनर्जी को यूज करती है, right? So that is about mitochondria. Okay, let's move on to the next cell organelle. The next is cell organelle. So I hope that okay. Yes. So now next cell organelles are plastids. Yes. The next cell organelles are plastids. Now another thing is that plastids are those cell organelles which are exclusively found to occur in plants. Yes, these are exclusively found to occur in plants. And in case of plants, that is these plastids, these are the second largest cell organelle, because in a plant, a central vacuole is the largest cell organelle, and after that comes the plastids, right? Now these plastids, these were discovered by the scientist E. Haeckel, right? So the scientist Ernest Haeckel, he discovered plastids, right? And they are exclusively found to occur in plant cell, right? Now plastids are again that is of two types. So one are called leucoplast, right? And another are known as chromoplast. Okay. Now this categorization is done on the basis that whether the given plastid contain a pigment or not. Means जो plastids हैं उनमें कोई pigment present है या present नहीं है. So that's the basic criteria for the categorization of plastids. Now first one is that is leucoplast. Now leucoplast, we say that these are colorless plastids. These are colorless plastids, means they don't contain any kind of pigment. I hope students you know the meaning of the word pigment with reference to science. Pigment means any color giving material in the field of science can be called as a pigment. कोई भी सब्सटेंस जो कलर देता है, जो कलरफुल है, किसी चीज को कलरफुल बना देगा, उसको हम पिगमेंट कहते हैं, राइट? So leucoplasts are colorless plastids, means they are devoid of any kind of pigment. They don't contain any pigment, so they are colorless plastids, right? Another is chromoplast. So from the term itself, we can take out the meaning. Chromo. The first part of the term is chromo, and the word chromo stands for color. Means those plastids which are colorful. So colorful plastids means these plastids, they contain a pigment in them, right? They contain a pigment in them, and that pigment is responsible for imparting them a particular color. So we say that chromoplasts, these are colorful plastids, right? Another thing, that most of your leucoplasts, they are found in underground plant parts. They are found in underground plant parts. Most of them, say for example, that's modified stems are there, rhizomes are there, roots are there, seeds are there. They all contain these leucoplasts, right? So generally, we say that leucoplasts, they occur under the soil, okay? Whereas most of the chromoplasts, they are found in aerial parts of the plant. Aerial parts means those plant parts which are occurring above the soil, like leaves, flowers, fruits, etc. And they all are colorful by nature because they are containing chromoplast. So we say that chromoplast, they generally occur in those parts which are present, which are comprising the shoot system of a plant, right? Another one is the main function of leucoplast is of storage. Yes, leucoplasts, they store different substances. They store different substances, and which type of substance they are storing, based on that they have got different names. For example, the plastids which store starch, we call them as that is amyloplast, right? Similarly, we have neuroplast, right? And then we also have alloplast. So amyloplast, they store starch, right? And neuroplast, they store proteins. And alloplast, they store fats. right? So what kind of substance they are storing, based upon that we have got different kind of leucoplast, right? Here that is chromoplast, since they are present in flower and fruits and just making the parts colorful. So their major function is photosynthesis, pollination, and seed dispersal. Yes, the three main functions of chromoplast are they help in photosynthesis, pollination, right, and seed dispersal. And I'm pretty sure you all are well acquainted with all these terms. So, for example, pollination. So, in low standard, you have learned about the petals, and we have discussed there that petals are the most brightly colored part of a flower, right? And that color is basically to attract insects and other animals to help them in pollination. Similarly, in seventh standard, you learned about the seed dispersal. So again, animals, they are attracted towards the different color, the attractive colors of the fruit. So they pluck them, they eat them, and seeds are dispersed, right? Now, with reference to that is chromoplast, we need to just have a look about the structure of that is chloroplast, right? So the chromoplast, special chromoplast, that is chloroplast. About this we should know. It's quite important. Chloroplast. So first thing, what is chloroplast? It is a kind of chromoplast only. No need to remain confused. प्लास्टिड्स आपके दो ही तरीके के होते हैं, ल्यूकोप्लास्ट एंड क्रोमोप्लास्ट, राइट? क्लोरोप्लास्ट, it is a kind of chromoplast only, and it contains a pigment called chlorophyll. It contains a pigment called chlorophyll. Now, since it is containing the pigment called chlorophyll, so we have given the name of that is chloroplast, right? Now, just let's have a look over the structure. Just give me 2 minutes. Let me finish off the diagram. Okay. So just I'm finishing your diagram. Okay. So outer membrane, inner membrane, that is again called matrix, right? You know, here just I have drawn this. So I have drawn like this. Yes. Okay. So this is called a granum, and these are called thylakoids, right? And this is called chlorophyll. Yes. And these are called threads. Okay. So that is the diagram for understanding. That is a diagram for understanding the structure of chloroplast. It is especially the diagram for the structure of chloroplast. Now, as you can see from the diagram that like any other plastid, these are also double membrane bound. Like mitochondria, plastids or chloroplasts also have double membranes, right? So that is outer membrane and this one is inner membrane, right? Now, the ground material, the floor of the plastid is again made up of transparent jelly-like material, which is given the name of matrix, or sometimes it is also referred as a stroma. It is also known as a stroma, right? Now, inside this stroma of this particular, that is a chloroplast, we see some small cells which are piled up together, right? So these piled up structures, these are known as granule, right? A singular term is granum, whereas a plural word goes as grana. Grana. The plural word goes grana, and singular structure is known as a granule, right? So basically, in all chromoplasts, these thylakoids are present, and these thylakoids they contain different pigments. Say, for example, in case of chloroplast, the pigment which is present here is chlorophyll. And about the role of chlorophyll, we have already discussed in previous question. Okay, I have already discussed in previous classes also that chlorophyll, it is a green color pigment, right? And its main function is to trap solar energy, and which is the main part for the process of photosynthesis, right? So here, that is this chloroplast, it is containing chlorophyll, and where this chlorophyll is present, it is present in the thylakoids. It is present in the thylakoids of the granule, right? Another thing, students, that like that of mitochondria, plastids also contain some ribosomes and their own DNA. Yes, their own DNA, so that is ribosomes. Yes, जैसे माइटोकॉन्ड्रिया में खुद के राइबोसोम्स और डीएनए फ्रेगमेंट्स होते हैं, इसी तरह जो प्लास्टिड्स हैं उनमें भी अपने राइबोसोम्स और डीएनए फ्रेगमेंट्स होते हैं. It means like that of mitochondria, plastids are also semi-autonomous by nature. So that you have to remember that the two cell organelles whose nature is semi-autonomous are mitochondria and that is plastids, right? Okay. And another thing is that I can see from the screen some of you are doing mutual conversation, so kindly try to avoid it. So please focus it here, right? Okay. So that is about plastids. And one more thing, children, do remember that is plastids. You have learned in lower classes that for plastids we call it as the kitchen of the cell. But the specific answer is these are the chloroplasts. जो प्लांट सेल में क्लोरोप्लास्ट होते हैं उनको हम कहते हैं किचन ऑफ द सेल. व्हाई? बिकॉज क्लोरोप्लास्ट कंटेन chlorophyll and that will help the plant to perform photosynthesis and that will lead to the food preparation for the plant. And what happens inside the kitchen in our house? Food is prepared. So chloroplast inside the plant cell, they are referred as the kitchen of the cell, right? And here from this particular plastid topic, question can be asked, especially from the types of plastids and differences between the two. So leucoplast and chromoplast, right? And do remember, chloroplast, they are containing chlorophyll pigment and they are kind of chromoplast only, right? दोनों के differences अभी हमने discuss किए थे यहाँ पर, so that are very important with the point of view of examination. कई बार पूछे जा चुके हैं और बहुत frequently पूछे जाते हैं differences between leucoplast and chloroplast, right? So that is about plastids, right? Now another one is that is the last phenomenon we are moving towards is that is vacuoles, right? that is vacuoles. Yes. Okay. Yes. So that is vacuoles. Okay. Now, vacuoles, if you remember in the class 7th, where we have studied the structure of an amoeba for the very first time, there you have learned that inside the cell are present blank looking bubble like structures which are called as vacuum, right? Sorry, which are known as vacuoles. Okay. Now vacuoles means that is empty. Means ये सेल वो सेल ऑर्गेनल्स है जिनको जब हम माइक्रोस्कोप के नीचे ऑब्जर्व करते हैं सेल में, तो ये देखने में हमें बहुत ही they look as empty, as if they don't contain anything, right? So those cell organelles are vacuoles, right? Another thing, these vacuoles are not double membrane bound. They have got one single unit membrane, and their membrane is given a special name that is known as stromoplast. Yes, the membrane of a vacuole is known as stromoplast. And the material which is filled inside a vacuole, it is in most of the cases, it is the cell sap. We call it as a cell sap, right? Now these vacuoles, in case of plants, these are generally large and single. Large and single. In most of the cases, in most of the plant cells, the vacuoles, they are large and single, and if it is, and in most of the cases, in position, it is central also, and that is occupying 50 to 90% of volume of the cell. That's why I told you in the lecture that in case of plant cells, these are the vacuoles which are considered as the largest cell organelles, right? So, in case of plants, vacuoles are generally large in size, central in position, and are single in number. These are one in number. Whereas in case of animals, these are small in size, scattered in position, and many in number. These are small in size, right? Scattered in position means in plant cell it is central in position, but in case of animal cells these are scattered, these are spread in almost every part of the cell. तो पूरी सेल में एनिमल सेल में ही बिखरे हुए होते हैं, so we call it as scattered, right? And many number, not only one, छोटे छोटे बहुत सारे vacuoles होते हैं वो एनिमल सेल में होते हैं, right? Now that is functions, which are the most important part for vacuoles. So first one is that is they provide turgidity to the plant cell. First function is they provide turgidity to the plant cell. You might have discussed about osmosis in previous part, in previous chapter, okay, previous part of this chapter, and there you have discussed that when we place a plant cell in hypotonic medium, then we find that the plant cell become turgid, it becomes swell up, right? But it does not burst up, right? So why does not burst up? Because of the presence of cell wall, of course, and along with that the vacuole which are present inside that plant cell, to some extent that also play. Means it when the liquid which is moving inside the cell, it exerts the pressure, so that vacuole in turn also exerts the pressure to balance it. So that is called turgidity, right? Another one is the vacuoles which are known as the food vacuoles. They help in digestion of food. I hope you remember the term food vacuoles in case of amoeba. In 7th standard, discussed that amoeba engulf the food particle, then food particle get trapped inside the food vacuole, and inside food vacuole, digestive juices are released. So there inside the vacuole, digestion is taking place. Digestion is taking place, right? Okay. So okay, from that RN, osmoregulation, that is also correct. Osmoregulation is a function of contractile vacuole, especially those organisms which are aquatic and are found to live in freshwater bodies, right? That is another function. तो जो organism की body में water का balance है उसको maintain करते हैं, right? लेकिन टर्जिडिटी मैंने आपको एक्सप्लेन किया, टर्जिडिटी का मतलब है जो प्लांट सेल्स की जो पर्टिकुलर सेल शेप है वो सेल्स को फ्लैसेड नहीं होने देते. That is because of the vacuole, right? Osmoregulation means maintain the level of water, concentration of water inside the cell of the organism. Okay? So let me write here another function, osmoregulation only. Now, osmoregulation, that is a function of the vacuoles, which we say contractile vacuoles. That is a special function of contractile vacuoles, right? Means they expel the excess amount of water out of the cell. That is osmoregulation, right? Another one is that is waste. Yes. Now waste means vacuoles. Some vacuoles they can store waste material, especially inside the body of plant cells throughout the life without harming the plant cells. That is first way in which they are helping. Second thing is that some vacuoles they also help in throwing out the waste material out of the cell, right? You might have discussed about that is endocytosis and exocytosis, right? So it is the exocytosis by which the waste inside the cell is thrown outside the cell. जो सेल के अंदर का वेस्ट है वो वैक्यूल के अंदर फील होता है और एक्सोसाइटोसिस प्रोसेस के थ्रू सेल के बाहर जाता है. So that is the waste removal, right? And finally, overall function of a vacuole is basically storage. Yes, the main function of vacuole is storage. They are known to store almost every kind of material in them, right? They are known to store every kind of material in them, right? Say for example, amino acids, proteins, sugars, glucose, water, salts, everything. तो vacuoles बहुत सारी चीजें अपने स्टोर करते हैं. So basically these are the storage houses of the cell, right? So that is about vacuoles, right? Okay, so students, okay, I can see some of the doubts here, but just because of time constraint, I would be noting your doubt, and in the next session, before bringing the topic, we'll discuss this doubt. So, do leave your doubts in that comment box because I always scroll down and note down your doubts. In the next session, we'll discuss this doubt also. So, just now I have to end the class here only. Okay. So, that's all about from my side for this particular class. And yes, that's all about the chapter cell and cell organelles. Right? So, in next session, we will be discussing some more topics. So, till then, take care. Bye.